Hi everyone, my name is Fritz Sedlazeck. I'm an associate professor here in Baylor College of Medicine in Houston, Texas. Today I'm presenting our work on structural variants map across 945 Han Chinese individuals using long-read sequencing. Before we dive in, this is a great collaboration together with Johan Fan from the Fudan University. So what are structural variants? Structural variants are 50 base pairs or larger genomic alterations. They cause a higher numbers of diverse base pairs between two humans than SNVs on their own, and so therefore greatly contribute to different diseases or different other phenotypes across species, but especially humans, as we are talking about them today. And it can be defined in 5 different variant types, being deletions, duplications, insertions, inversions, and translocations. Traditionally, we detect these variant types across mapping of short reads or long reads, where we identify abnormally distant paired end reads from short reads or abnormally split reads in short reads or long reads. Split reads are with 2 or more segments of the same read are mapped to different locations. Over the past years, my group, as well as other groups, have identified long reads really being superior in identifying structural variants compared to short Illumina reads. They're usually more comprehensive, so for short long reads, we usually take 20 to 23,000 structural variants in healthy individuals. For short reads, we only often detect 10 to 12,000 structural variants, mainly missing insertions and having a large false positive identification. Long reads also enable the access in repetitive regions and are highlighted here in 2 or 3 works already as being important to assess also medical relevant genes such as LPA, HLA, which we are almost blind to with short reads on their own. And they also contribute to better assemblies, for example, the T2T work that is currently going on. So therefore, it's no surprise, as we highlighted in our recent review, that the amount of studies that are using long reads are increasing over time. So here time on the x-axis and the number of samples per study on the y-axis. And we indicated here with the circle the genome size that is targeted. So back in 2018, before all this mess started with COVID, I visited the Fudan University to start a collaboration, start discussions with them, with Johan Fang's group. As you know, Han Chinese are the largest ethnicity, around 1.3 billion people. There is no structural variant catalog available, published even yet, and there are few to non-reference style genomes assemblies. So one of our really goal targets in the studies was to improve the understanding of structural variants and their impact by sequencing 500 individuals initially, and then we extended it now to almost 1,000 to study evolution, but also study the impact of structural variants on the medical field and give a structural variant catalog. We achieved this by leveraging the Oxford Nanopore Promethean and sampled, and all the samples came from this one district. To keep them comparable to each other. What we do for structural variant calling itself, well, we used sniffles, one tool that I developed or my group developed over the past years together with NGMLR, which is the long read mapper. So we mapped those reads from Promethean directly to GSH38. We then used sniffles to call structural variants initially. That has around the sensitivity of 88% and the precision of 85%. When compared to genome in a bottle, this standard. And then we used Survivor Merge, a tool that also I developed over previous years, to combine these single sample structural variant calls. As you can see on the bottom right, there's a distribution of coverage that we achieved. This were kind of early nanopore data sets on biobank samples. So we see that we cut or threw out samples that didn't reach a coverage of 10x, and you see that our average mean coverage is around 15x. So to achieve still a high sensitive mapping, what we did next was to re-genotype. So we took the file that Survivor Merge produced and re-genotyped that file across all samples using sniffles. And what this improves is the numbers of structural variants called per individual. However, this only, of course, works for common SVs. So an SV has to be observed first to be re-genotyped in other samples. It has a massive effect. So when we do the raw calling, as we call it, you see the green points across the samples, which is indicating the number of structural variants and giving coverage where we see a clear correlation, right? When we now do this kind of re-genotyping with sniffles, we can re-identify structural variants much easier. And that leads to this red brownish representation of number of structure variants per sample itself. So this was a great success and just enabled by kind of clever software implementation, so to speak, from my group. So how does this structure variant corset look like? Well, here on the right side, you see the standard SV comparisons and length comparisons. So we see this very dominant ALU peak of insertions and deletions in green and in orange. We see also smaller SVA and line peaks as we would expect them. Overall, the structure variant corset is very well defined and then follow the expectations. So we have mainly deletions and insertions as structure variants, followed by duplications, a few translocations, and very few inversions, 2.6, 1.7%. What was interesting for me to observe was the different levels of deletions versus insertion rates. When we often look in literature, insertions are often defined as a little bit higher than deletions actually. However, on the population scale of the Han Chinese, we don't observe that directly. But when we zoom into an individual scale, we see that the inversions are indeed higher than deletions itself. And I just see I have that labeling wrong here, sorry for that. So this follows kind of the expectations that, for example, we know from 1000 Genomes Project where they assembled individual numbers, but never looked at the harmonized population set. So here it makes sense that insertions are evolutionary longer, persisting already in the population, might accumulate additional mutations that we would have currently merged together irrespecting certain SNVs in the insertions, and that is a research topic that is still going on. So therefore, in the allele frequency, we see a distinguishment of insertions that are in general more common than in deletions on their own. In terms of genomic locations, we see the general standards, as we would expect, more intergenic than exonic, impacting structure variants. What we observe by comparing it to other data sets, we see that 52% of our SVs can be named as novel SVs that have not been observed before due to the great resolution of long reads, but also due to the deep study of these Han Chinese data sets. We also genotyped these structure variants from the 1000 genomes, or the HGSV, across 100 Illumina data sets to just kind of have a comparison and quality check. And as you can see on the plot here, the allele frequency versus the genotyping rate. So if we take rare SVs from our data set, we often have a low genotyping rate, whereas if we take very common SV to almost fixed SVs from our data set, we have a high genotyping rate in the 100 Illumina samples. And this is exactly what we would expect and indicates the high quality of our data sets, besides other QC checks. So the other interesting point was that actually we see a higher agreement between independent long read studies versus this large cohorts of GNOMED SV or initial 1000 Genomes project. And one of the things we think this is point here is that these short read large studies are often missing something very systematically, whereas the long reads are having a better characterization across the genome and therefore also detect variation structure variants. So therefore we have a high overlap here. One very interesting fact, and sorry about the time, I'm just like trying to show you multiple interesting aspects about the study, but the paper is currently in review, so we'll be out hopefully soon. It is one of the interesting aspects is of course that we identified multiple structure variants that have clear implications in certain pathways or in certain genes. So one of these genes here is, for example, the AMY locus, which is a well-known locus and for human adaptations of high starch diets. So rice and wheats do have high starch grains and are, of course, the main food source of Han Chinese. We found that AMY locus is a specific SV hotspot in our study, although 2 NGS-based studies all included hundreds of samples from Han Chinese haven't seen that before. To go further, we also looked at natural knockout genes. So those are genes that have a deletion of a high frequency in our population data set. So we identified 15,000 homozygous deletions across, but only 300 of them are overlapping with protein coding genes, and 219 of them were not previously reported. And I'm just listing 3 of these genes where we identified knockout genes. So 10 knockout genes we identified that had an allele frequency of the deletion and greater than 90%. So across the population of Han Chinese are pretty knocked out. In singles, like 14 CNF98 and CNF192 are just a few representatives of that. Another interesting finding that we had was this small 172 base pair deletion that you can see here indicated by these black bars. I hope you can see my mouse. In the ACAN gene. The ACAN gene is a known gene that encodes or has an impact in human heights. So when we look this gene up in the EBI genome GWAS catalog, we see multiple SNP hits across this gene. So we know this gene is relevant for human height or body size. And we just labeled them here. You see here multiple heights or body size indications. When we stratify our data set according to this deletion, we can see a little bit of a trend. It's nothing too significant, frankly, but it's a nice trend where we see like people that having a homozygous deletion at this locus, the blue group, have a little bit of a lower height than the red group that don't show any deletion. And we see that people that have a heterozygous genotype at this locus is somewhere in between these two groups. Another interesting find was this 51 base pair deletion that overlaps an enhancer at the intronic regions of AATK, which is also an interesting locus responsible for height and body weight. So this here, as I said, overlaps the enhancer, not the gene directly. However, we found multiple hits indicated down here with a connection map that connects these two loci together. AATK and BAP2 have implications across multiple phenotypes, but as I said, one of which is body weight as well. And when we stratify again our data set, accordingly, we see again like a little bit of a trend. However, this time it's less clear because the group in red that doesn't carry this deletion is kind of overlapped with the group in blue. But if we focus on the median, the black lines here, we see a little bit of a trend. It's nothing significant, but it was quite interesting to identify that as well. So in the last few minutes that I have in my talk, one thing that we really wanted to also look into was what we are missing in the current reference genome, being it either GRCH37 or being a GRCH38. And as you know, there are also now currently multiple groups working, including my group, on the T2T reference genome, which is the CHM13 version of a human genome and has included multiple new regions along the genome to resolve not only telomeres, but also centromeres itself. So to do that, what we did, we took all the reads that were initially unmapped by NGMLR and tried to assemble them. So after the assembly, we could stratify these contigs out. So 851 of these contigs mapped actually back to GRCH38. Might have been just like repeats or across different N regions of the genome itself. 1,478 mapped actually nicely back to T2T, as we can see in the next slide, in certain interesting regions. 700, it's almost 800 contigs mapped to hominidae, which I'm going to discuss a little bit further. 372 could still be anchored, however, across almost 1,800 of them. We don't really have a good sense what they belong to right now, and could mean that they are Han-specific novel sequences, larger insertions or larger novel sequences in telomeres or centromeres, but also could be other things. The important thing to show here, we also screened, of course, for contamination based on the assemblies, based on the sequence preparations or anything. So it's not bacteria, it's not like the typical things that you would find as contaminations. This has been all taken care of. So as I said here, on the right side, you see some connection maps of where these extra contigs are mapping. So in the T2T, it's obvious it's going towards the telomeres and some of the centromeric regions. Same on GRCH38, it's close, it's paracentromeric regions where we often see ends in the genome right now in our human reference. The most interesting part for me, however, is this hominidae. And so I'm showing you just here, a cherry-picked, of course, one region. So here is UCSC browser. In black, we have the contig that we assembled. In in brownish here is the assembly of the chimpanzee genome, and we can see that we map this contact from the Han Chinese population onto the chimpanzee genome. This, of course, doesn't indicate that we are studying chimpanzees here, but has the interesting effect that this contact branches over a region that is indicated as a deletion in humans. So this means that the reference here is certainly biased and doesn't well represent the Han Chinese population, and that this contact should be used in this reference region itself to complete the reference, because this deletion is apparently not true in GRCH38, right? So to summarize my talk, what have I shown? We could show novel structure variants with the utilization of long reads. We could show long reads being cost efficiently used and streamlined used in large population studies. We could identify interesting phenomena around insertions to have a higher allele frequency than deletions, maybe because it's in the same population where we are studying them compared to deletions. We could see interesting effects of the structure variants hotspots, as well as phenotypic impacts across this study. And in the end, I briefly showed you some examples where we could see how we can extend GRC38, but also T2T itself, and how much this is needed as a medical or clear human represented reference. For sake of completeness, these were the mainly methods that we are using, NGMLR to map, sniffles to cause structure variants, which are both published in Nature Methods, Survivor to merge and compare structure variants and to distill summary statistics of structure variants into Locus, which was published in Nature Communications. And with that, I want to thank, of course, my collaboration partners, and I hope I can visit them very soon after this whole COVID mess is over at the Fudan University in China, as well as, of course, my own group and my center that I'm working on, the Human Genome Sequencing Center at Baylor College of Medicine, and NIH for generous funding. If you're interested in this kind of work, please feel free to reach out to me either over email or just directly message me over Twitter as well. Thank you so much for this opportunity to present our work today, and I'm happy to take any questions.